Good morning, good afternoon, everyone. I'm Angela with ProFarm, and I'm pleased to welcome you to our webinar today on preventing disease and improving IPM efficacy in grapes. All right, today's speakers, I'm pleased to introduce you two of my colleagues. The first is Robert Blundell, and he is a microbiologist and plant pathologist that joined ProFarm Group about 2 years ago. Robert has a wealth of experience, about a decade, in plant pathology, and more recently has spent about 4 years in viticulture. He originates from the UK and received his bachelor's of biological sciences from the University of East Anglia. And then he actually came to the US and received his master's in plant pathology from UC Davis. His career spans nearly a decade, and he has experience from working as a laboratory technician and then a graduate student researcher in plant pathology at UC Davis. He also most recently worked with Colorado State University as a viticulture extension specialist, something I didn't know. Colorado's growing grapes, and as you can imagine, the conditions are quite challenging. So Robert has a lot of experience in how to mitigate economic losses and develop best management practices in grapes. So he's going to be a wealth of information and really go through kind of today's modern issues on vineyards and share some best practices and suggestions for getting ahead of disease. Then, I have my colleague Jake Wiley, who covers the Northern California territory, and he joined ProFarm Group 1.5 years ago. He not only covers Northern California, but he also covers Nevada and Hawaii and works hand in hand with growers and PCAs to improve and develop new approaches to IPM programs in both organic and conventional crops. Before ProFarm, Jake was a crop consultant and a PCA for Nutrient Ag Solutions, where he managed disease, pests, wrote recommendations for rice, tree nuts, tomatoes, and grapes, and managed both soil health and fertilizer for customers. So Jake has his Bachelor of Science in Agriculture, Crop Science, and Horticulture from the California State University, Chico, and he is a licensed pest control advisor and a certified crop advisor. So Jake is going to be talking a little bit about the different IPM programs that he would suggest based on Robert's discussion about modern pest problems. Before we get into the content of our webinar, I just want to remind you that ProFarm Group is a publicly traded company, and any information that is shared in this presentation is strictly educational. Should you want to invest in our company, please do your own due diligence. All right, so just a quick overview of what we're going to talk about, and then I'll hand it over to our first speaker. So we're going to go over the current and future challenges in vineyards. We're going to review best practices for grapevine trunk diseases and best practices for managing mealybugs. Now I'd like to hand it over to my colleague, Robert, who's going to dive deep into many of the things I'm sure you're probably thinking about as you get ready to start the next crop year. So Robert, I'm going to go ahead and hand the slide control over to you.
Perfect. Thank you, Angela. Thank you for that introduction, and welcome to everybody on the call today. It's a pleasure to be able to speak to you. It's been a couple of years since I've kind of done any viticulture webinars, but it's still a topic that's very close to my heart. So yeah, I'm very excited to chat to you today. And kind of when I was asked to give this webinar, I was kind of given free rein to an extent. So I've tried to pick not just diseases, but also a few other pertinent issues, including cold and heat damage, as well as a few emerging pests. So that way, wherever you're kind of joining us from in the US today, there should at least be one topic that's of interest to you. So kind of before we get into the heart of my talk, I figured we'd keep you on your toes early. So as Angela said, we have a question. And oh, let me just pop back here. So yeah, as she said, if you could put the answer in the chat, and it'd be the first person to correctly answer. And my question is, what caused this? This was a photo I took in Western Colorado about 2 years ago. And I believe it was in about May. And this is a split vine as a result of excessive winter injury. So, and Danielle as well, they're awesome. So that, yes, actually a severe frost injury actually technically would be kind of more correct, as not always do you get a split vine. But in this particular case, it was a very severe winter injury. Awesome. And then, yeah, just a quick overview of this section of my talk. So this is kind of current and future challenges in the vineyard. So yeah, really it's going to be abiotic stresses, so the cold damage and the heat damage. A topic, particularly as a pathologist, that I'm interested in, so is vineyard sanitation, and that kind of goes hand in hand with mitigating disease complexes and inoculum buildup. And then we're going to have a kind of emerging pest case study before we get on to some of the best practices for disease and pests. And kind of thinking about abiotic stresses, particularly with mitigation, we have as a common theme, there's going to be short-term management and long-term management that we delve into as well. Fantastic. Okay, so yeah, so when it comes to cold damage in grapevines, this is when the temperature drops typically below the cold hardiness value of a grapevine. So all grapes have a varying range of cold hardiness depending on their cultivar. And essentially what happens is we're going to get bud and vascular damage. And as we saw in the quiz question, to a severe extent, you could also get trunk damage. So the buds and vascular tissue, they are the most sensitive, and kind of in the top two left photos there, you can see the kind of the makeup of a bud. So grapevines have compound buds. So we have the primary there in the center with the secondary on the left and the tertiary on the right. And on the left, we're seeing kind of a healthy bud. So we're seeing green tissue that tells us that the phloem and xylem's are living. Whereas on the right, we're seeing brown and black tissue indicating some form of damage there. Yeah, so as I said, so buds are kind of the first point of injury, but also this will extend to the canes and also throughout the vine, definitely through the trunk and potentially even down to the roots as well. So in the slide here, we're going to cover kind of what is cold damage and then understand what can we actually do, to kind of mitigate it. It's hard to ultimately prevent cold damage, but there's certainly some good steps we can take in the short term and the long term. So cold damage can really kind of occur at 3 different times in your vineyard. So you can kind of have pre-acclimation cold damage. So that's before the grapevine has had sufficient time in winter to harden off. So all grapevines, as I'm sure you know, enter dormancy during the winter. So if we say you get a cold event around maybe September, October, that kind of comes out of the blue, that's a very common period to get some cold damage, as that time of year, they really haven't had enough time to start hardening off yet. And then another time is actually during winter dormancy. And this may just be the result of an extreme cold event that basically pushes the temperature to below the actual cold tolerance. So, they've achieved their full potential for cold tolerance, but, the temperature is so low that they can't keep up with that. And then spring frost damage is the final one. So, this may be kind of any time from maybe February onwards, but it really kind of depends, where you're growing grapes in the country. Obviously, things warm up quicker than other places. But this spring frost damage typically occurs when you kind of have a warm event followed by potentially a frost, or not even a frost, just a particularly cold event that will throw them into damage there. So yeah, when it comes to actually identifying whether you have cold damage, firstly, you're going to definitely want to look at your buds. So you're going to want to make a careful set of cuts, essentially to expose your primary, secondary, and tertiary buds. And as a result, you're going to be looking for that greenness. So, the primary bud gives you the majority of your fruitful crop for the next year. So if you have a healthy looking primary bud, then you're hopefully in good shape. If it's not looking so great, you're going to want to move on to your secondary bud. Secondary buds are not great when it comes to producing a crop, but they can somewhat give you a little bit of lease, and the tertiary is more of just kind of a survival mechanism for the plant and doesn't really give you much of a fruitful crop. So when it comes to now understanding what to do with this, once you've looked at that, so if you do have extensive primary bud damage, the number one thing you're going to want to do is adjust your pruning accordingly. So basically, if you do have, very severe damage, you're really not going to want to prune too much. And typically per state, there are recommendations based on the percentage of primary and also the secondary bud damage of how much pruning you should be doing. And basically the idea there is to, allow some sort of buds to remain to hopefully, give you that following crop. And definitely it's important to do representative sampling as well. You're not just going to want to pick, say, 5 vines on the same row in one particular part of your field. You're going to want to take it from different parts of your site, particularly if your site has a change in elevation, as we see even very small microclimates exist, especially with elevation changes. So that's going to be important to kind of check accordingly across your vines, but definitely per cultivar as well. As we mentioned, there's different kind of levels of cold hardiness. So yes, once you've been able to identify that bud damage, you're also going to want to kind of check the health of the rest of the vine. So you want to cut open your canes, and hopefully you're going to see some green tissue in there, again, to suggest that the phloem, as well as the xylem, the water conducting tissue, is still healthy. If you're seeing the black and brown tissue there again, you're going to want to look further down to your vine. And hopefully you're not going to see a huge crack, as we saw, kind of extending down your trunk. And typically, if you do have kind of extensive vine damage, that's going to manifest itself in spring, and that's going to be because it's going to produce suckers, so green shoots coming off the base, and that's kind of a response to that severe damage. However, that those suckers are basically, showing that you do have a chance of therefore regeneration. So if you're doing self-rooted vines, you're going to want to cut it right back to the base of the ground and then start training those new suckers up. And you're going to want to keep a couple of those, as again, you may be having future cold events. See if this basically is increasing your chances of survival. So train a couple up and keep that strongest one going. If you're using grafted, that's a little bit more tricky. So you're going to want to cut all the way down to just above the graft union. And then you're going to wait for your sucker production. And if the suckers are coming from above the graft union, you're okay, you're in business. That's going to be your scion that's perfect, that's going to regrow there. However, if the suckers are coming from below that graft union, that's going to indicate that the scion is dead. And if you're going to want it to continue to grow that grafted variety, you're going to have to replant there. So kind of, yeah, retraining, that's really the biggest thing to do if you do have extensive cold damage. And then there's also, fortunately, some preventative things that you can do as well. So the second point there I have on short-term management is hilling up. So this is particularly useful if you do have grafted vines, and hilling up is essentially the practice of mounding soil kind of around that graft union. You're going to want to do this well in advance of winter or any kind of full cold events you might typically experience. And the idea there is just to increase kind of the ambient temperature around that graft union just enough to prevent any extreme damage. Other options available. So we have wind turbines. These are expensive, but depending on how often you're experiencing this and kind of the type of weather events you do have, that can be very useful. So kind of wind turbines are very useful during inversion temperatures. So basically when you have a layer of cold air stuck below a layer of hotter air, and by having the wind turbine, it's going to mix those layers and hopefully increase the temperature just around your trellis height, and kind of higher than that, enough to, again, alleviate some of those really severe effects of the cold. And then, yes, another thing, so we mentioned retraining new suckers, and the next one is kind of going to be a theme of at least my portion of the talk today, and that is just, having good general vineyard practice is going to be key to mitigating some of the damaging effects of, say, cold damage or even heat damage, because a healthy vine, so via, good irrigation, good fertilization practices, as well as good disease management practices, are really going to make a difference when it comes to when the vine experiences these cold events. A diseased vine is going to be somewhat compromised, or a poorly irrigated vine may not be able to enter dormancy, as well as, say, a fully like a well-irrigated vine. So, just having general, and these more the simple things, general good vineyard maintenance can actually go a long way when it comes to that. And I just wanted to highlight the bottom photo on the left there. This is actually a photo from France, I think 2 winters ago. And this is in the region of northern France, where they actually, they sometimes light heat sources essentially in the vineyard to, actually just raise that temperature. And so it's quite some extreme lengths people will go to. But yeah, it makes for some very dramatic photos. And that final photo on the bottom right, that was actually a vineyard in Colorado from the same kind of winter event that caused the quiz photo, where again, the grower just had to come along and completely chop out his vines back to the ground again. All right. So now, kind of inversely of cold damage, rather like, extreme cold, grapevines are also very sensitive to extreme heat. And when we're talking about heat, we're essentially talking about heat waves. So typically by definition, you're looking at about, 3 days or more of over 100 Fahrenheit for it to be considered heat wave or an extreme heat event. Grapevines, fortunately, are fairly heat tolerant, but even they cannot, withstand some of the extreme temperatures we do see creeping into some of our summers or full temperatures now. So in terms of kind of the negative effects that you will see on a vine as a result of extreme heat, first you're going to have excess evapotranspiration. As we sweat, to release heat, the vine's going to do that, but essentially when it's doing this, it is really dehydrating itself. And so with that combined with, say, stomata closure and then a reduction in photosynthesis, that is really just kind of shutting the vine down. And actually, as a last resort, the vine will draw fluid, so water, back from the cluster, back to its vine, essentially as a survival mechanism, so it doesn't die. As a result, you're going to get shriveled clusters, reduced yield and quality there. Kind of thinking about quality as well, excess heat and UV can reduce the anthocyanin content of your grapes, so reducing the quality there. And it can also degrade that wax layer on your berries as well, so it's leaving them a lot more susceptible to rots. And kind of under mild heat events, you'll see some kind of brown necrosis scarring. And actually under severe heat events, you might get full necrosis, which then leaves the berries, susceptible to kind of a lot of bacterial diseases that may come in and kind of rot your berries, as well as also kind of attracting pests. However, again, like cold damage, there are fortunately some techniques that we can implement in our vines to alleviate heat stress. I should mention that heat stress has really kind of become a focal point, I would say, particularly here in California in the last 2 to 3 years. There's a lot more research being invested in this now to understand that. And a lot of the grape days that I've seen the agendas for, they're incorporating this. So firstly, irrigation. So irrigating prior to a heat event is going to be key to kind of prep the vine for that. If you wait to irrigate during the heat event, it's probably going to be too late. So definitely advanced planning there. One big thing, and this is a topic that can be incorporated for disease control as well, that is shoot thinning and leaf removal management. So basically an exposed cluster with a very little leaf coverage will be considerably hotter than say maybe a more shaded one. And I think about up to 15 Fahrenheit even has been shown to be a difference between those. So you're definitely going to want to kind of manage that leaf and shoot removal. That being said, that may affect the prevalence of some diseases. So it's a little bit of a challenge kind of getting that right. Also bare vineyard floors. So if you have bare soil, that temperature is going to be considerably hotter than say if you have a cover crop growing there or something else. So that might be something to consider. Trellis type as well. This kind of fits in with short and long term, because if you're starting a new vineyard, you're going to be wanting to think about trellis type right from the get-go. So VSP, vertical shoot positioning, a very popular one. However, it can result actually in more sunlight exposure. There are advantages of having more exposure, but in the case of heat damage, that's going to be potentially negative. Foliar applications are also an option. So there are some products that exist that essentially act as kind of a sunblock. So kaolin clay is a popular one. Kaolin's also used in our sunscreen that we apply on our bodies. And the idea would be to apply it foliarly, and it basically will reflect the light. So it's keeping some of those harmful UV rays off, again, just kind of to hopefully bring down the temperature just low enough to mitigate some of that extreme heat. But with that, you're going to want to apply those before a heat event. Again, not during it. It's a little late at that point. So it's, kind of key preparation, and vigilance is key for these abiotic diseases. And then lastly, for kind of management for the short term, some of the ISR, SAR products, they can be very beneficial. So again, we kind of talked about good vineyard management practices. So applying some of those that can kind of potentially boost the health of the plant, whether it's defense responses, against diseases for that season, will become very beneficial again. Because if you already have diseases in, then your heat damage could be a little bit more severe. So in terms of long-term management, you have a couple of options, and they mainly focus on site selection and row orientation. So, with cold damage, with site selection, you're going to want to have sloping sites ideally, because cold air, sits in valley floors. But if we're thinking about heat damage, it's really the orientation or the aspects that we're thinking about. So north or east facing slopes with that east-west row orientation will actually can actually make a big difference in terms of a little bit of relief from the sunlight and extreme temperatures. So that can be something to think about. And then the other big one is cultivar selection. So with that, you may want to pick a early ripening variety if you're, say, having, extreme heat events, perhaps later in the summer, whether that's August or September or closer to harvest. You may want to consider cultivar selection to kind of get ahead of that and beat that. And that kind of goes in hand with dealing with cold damage as well. Apologies, I forgot to mention. So, with long-term management of cold damage, cultivar selection is key. So, vinifera is going to typically do very poorly with extreme cold, whereas hybrids are, they have a better genetic makeup essentially to deal with that considerably better. And more and more wineries, vineyards are adopting hybrids these days. if you live in an area where maybe you're experiencing these extreme events not as commonly, you may want to think about doing it a little bit. But if you're, for example, where I was a viticulture specialist in Western Colorado, it was a real challenge for anyone to grow vinifera, and they pretty weren't making money off of it. So for them, it was definitely a safe bet, planting hybrids, and hybrids as well. They've had this kind of reputation for a long time that they don't produce as quality wine as you may want from vinifera. But winemakers are certainly understanding more and more these days how to make great wine, which I've had a pleasure of experiencing. As well as they can also be a little bit more resistant to some diseases as well, which is another benefit. So moving on to vineyard sanitation. So this is kind of the topic, combining mitigating disease complexes in inoculum buildup. And why are we kind of interested in mitigating disease complexes? Well, pretty simply, multiple diseases are worse than one, and there's a lot of disease complexes that do exist for a lot of crops, including grapes. And also it can be very hard once you have a disease complex to identify, the main cause, and therefore it's tricky to kind of take actions to therefore deal with that when, there's multiple in play. And again, so minimizing abiotic stress, it's a great place to start. So again, irrigating sufficiently, fertilizing, you're taking some of these steps to, maybe prevent against cold damage, heat damage, just the general health of that vine is going to be key. And I will, keep kind of reiterating this throughout the talk today. And then, really your three main disease complexes you might be concerned about in grapevines are going to be nematodes, viruses, and fungi. So for nematodes, rootstock selection can be key, whereas if you're trying to reduce viral load, you're going to want to have certified virus-free material kind of from your nursery stock. Fungal diseases, that's kind of where I spend a lot of time working with fungal diseases. These are a huge issue in vineyards, and there's kind of a couple of reasons for that. But mainly, number one, they are very specialized at overwintering. So they produce these specialized structures that can basically survive the winter, and they will be present on, various parts of your living vines, whether that's on the bark, the cordons, the canes. But importantly, they can also survive on, say, pruning and harvest debris as well. So once you've gone through your vineyard, you're creating a lot of debris at pruning and harvest are those two key times of the year. So it's very important to do your best to kind of reduce that inoculum load. So whether that's burning, chipping, or even sanitizing, those piles will be key. And again, it is a lot of manual labor, it's a lot of time investment, but just little things like that can really help you in the long run, just mitigating those diseases, because again, they're just providing that inoculum for the season to come and seasons down the road. Removing vines with canker pathogens, virus, and pests. So yeah, this is kind of the step you're going to want to take. Maybe you've tried some things, tried some strategies, but you have very heavily infected vines, whether that's a virus or some of the trunk disease pathogens. You may just want to remove that vine from your vineyard and just completely remove it as an inoculum source. Again, it's hard to cut out some of these vines, but in terms of the longevity of the rest of the vineyard, this can be very beneficial. But you're going to definitely want to make sure you've kind of diagnosed your disease first and check it's not an abiotics issue that you could be solving via fertilization or nutrition, for example, before you kind of go in and cut those away. Sanitizing equipment. So a lot of the oxidizing products available, maybe some of these were initially developed for, say, cleaning irrigation lines, for example, they can be very effective. So, spraying down key bits of equipment, whether that's pruning shears, if you're using mechanical pruner, those parts, moving parts in your vineyard, your tractors, your other moving equipment. But not just that, you can spray, say, your burn piles, or instead of burning, you could be spraying those piles as another option there. And that kind of goes hand in hand with vineyard worker awareness. So just kind of having everybody up to speed on the fact that, inoculum is present in the field. So, if you have a very heavily infested site, say one of your 10 sites, for example, you won't want to go to that site first in the day. You're going to want to go to there last. You may want to be sanitizing boots, things like that. Just small steps that could be made, especially if you're perhaps sharing equipment with other farms. Just kind of know if your farmer has a disease, and then you may want to consider spraying it down, for example, before using it. So yeah, it's it's a tough challenge to mitigate disease complexes. They're just prevalent everywhere. But there are certainly some, in theory, these are simple steps, but I definitely understand and empathize that it is challenging and time consuming to kind of take a lot of these steps, but a few of these should hopefully help in the long term. All right, so moving on to kind of the three main best practices and problematic pests, so the mealybug, the powdery mildew, and grapevine trunk diseases. Before we get into that, I just wanted to do a quick case study of one emerging pest, as kind of the theme of this talk today as well was, current as well as future vineyard problems. So I figured I would cover spotted lanternfly, and this only came to my attention a couple of years ago by means of a extension bulletin kind of warning people about it. So spotted lanternfly, so this is characterized as a invasive plant hopper, and it's really been spreading mainly on the East Coast. I believe it was first detected in Philadelphia in 2014. I think I saw there was somebody from Philadelphia on the call today, so I'm glad might be a few people this is affecting so they can think about this. And I think it's, spread to about 14 East Coast states to date. So it's a phloem feeder. So it's basically capable of blocking that sap flow in the vine. So it's really then shutting down the foliar tissue in the upper aerial parts of the vine. So it's kind of starving them of essential nutrients. It also deposits honeydew, which basically serves as a substrate for sooty molds. So again, that's going to cause contamination in your clusters kind of down the line. So with the spotted lanternfly, eggs are mainly laid in September through November, and they hatch in April to June. And before the adults emerge, there are basically four nymph stages. So when it comes to control, detection is really key. So you can see in this photo on the left, we have this kind of large egg mass. So that's indicative of the nymphs that are going to emerge. And then on the top right there is the kind of the adult with the characteristic black spots. They often don't actually open their wings fully. They're not huge flyers. They typically actually just move more via movement as well as transit, which is how they're actually getting spread a lot of the time in shipments, things like that. So yeah, ideally you're going to want to detect those. If you can remove that egg mass initially, that's going to be hugely beneficial at reducing the eventual adult emerging population. One good thing is that the lanternflies they do quite like to occupy the vineyard edges, as that's where they're coming in typically from surrounding horticultural crops. So a good place to start is going to be that vineyard edge to kind of limit your the area you're going to have to search for. One great technique for kind of controlling is to detect as well as remove those key plant hosts. So the tree of heaven is kind of the iconic host or spreader really, or spotted lanternfly, as well as a number of other horticultural crops. So if you can identify those and find that you're getting populations in those, you may just want to remove those from around, kind of secure your vineyard borders, as it were. Netting can definitely be an option. It's a little bit frustrating in terms of then getting access, into your canopy, but certainly physical netting is just a great way to try to keep out these pests. Definitely applications are available for nymphs and adults. There's definitely a lot of ongoing research still kind of into what insecticides are going to have the most effect. And typically, with insecticides, you're going to want to hit the population early before all of the adults emerge to have your kind of best chance with that. So I know there's a lot more ongoing research around the US right now into kind of foliar applications that can be made. And then, I wrote there, questions remain over the effect of spotted lanternfly on plant hardness and susceptibility, like kind of all other pests and pathogens. They likely have a negative effect on, the general health of the vine. So, again, so you're going to have a lot worse, maybe cold damage or heat damage if you have an infection. So like all things, they're all kind of tied together, those abiotic and biotic stresses. And before anyone kind of west of the Rockies or west of there's a lot of people in California on the call, it is anticipated that spotted lanternfly will probably reach the West Coast. I think I read by about 2027. As I said, it does spread very easily, whether that's on, say, trucks for shipping, things like that. So it's definitely making its way across the country, and its presence is known and is definitely is a very large concern for the viticulture industry going forward. Okay, so grapevine trunk diseases. This is a favorite topic of mine. I spent a couple of years doing some master's research on this. Trunk diseases are a huge issue for viticulture in the United States, as well as pretty much anywhere that a grape is grown around the world. And basically, it's due to the huge number of fungal pathogens that are responsible for trunk diseases. And trunk diseases, the best kind of term, it's a very umbrella term, I should say. So on the top left of my slide there, you have Botrys dieback, E-type dieback, Esker, as well as Phomopsis. They are kind of maybe the four key ones. And underlined there is Botrys and E-type. Those are two very major ones, and ones that I did some work on here in California. And particularly with Botrys dieback, there are actually 23 fungal pathogen species known alone just to cause, that single type of trunk disease. So again, they are very, very challenging to control. And kind of as well as, there's a lot of species that are causing this. The main challenge is that really these fungal spores are entering via pruning wounds, as well as nursery wounds to kind of a minor extent. But obviously, everybody that has a vineyard is going to be doing pruning. Some of these huge vineyards, that's an insane amount of pruning wounds available. It's just, it's kind of an open playing field really for these pathogens to enter in. So they're vascular pathogens, so they're going to get in your pruning wound, and then they're going to occupy the phloem, the xylem. They're going to move pretty much throughout the vine in that moving tissue. So in the bottom photo, you can see a cane we cut open a couple of years ago, and the dark shape on the left, that's the presence of the pathogen there. So we refer to these as kind of canker pathogens. And then on the right there is actually one of the fungal species that we isolated just from sterilely taking a few cuts of that, so grown in the lab there. So as I mentioned, yeah, they form these characteristic cankers. So your type of dieback actually forms typically wedge-shaped cankers, whereas Botrys are a little bit more irregular. So in terms of some of the diseases, grapevine trunk diseases are actually fairly easy to identify in theory. The top left photo shows the extreme dieback on one side of the vine as a result from that. So as I said, they're vascular pathogens are shutting down the transport up to the top part of the vine. So yeah, typically with E-type, you get dieback of one of those arms. So as well as that, you're going to get delayed bud break, chlorosis, and as I mentioned, the stunted dieback. So kind of similar to some of the other fungal pathogens, they are very specialized at producing these overwintering fruiting bodies. So trunk diseases produce pycnidia. So in the central kind of top photo that's highlighted in red, you can actually see with the naked eye, you can see these pycnidia encased in stroma. They're very visible, and that's what's going to be your kind of inoculant source for the next year. Fortunately, there are quite a few options available to us with trunk diseases. However, they need to be preventative and protective practices. It's very hard to cure a trunk disease. So as I mentioned, pruning is the main entry point for these trunk diseases. So that's kind of where, like cold damage, we're going to first focus our efforts on. So the graph on the bottom kind of highlights that the incidence of a trunk disease inoculum release is tied to rain. So particularly there in the winter, January, February, March. This was a study done in Sonoma County, I believe. So pruning in dry conditions is absolutely essential. Any kind of precipitation, not just rain, it can be fog, it can be dew. This all contributes to a huge spore release. So they're present on the bark, they're present on the canes. So as soon as they get released, they're already in the immediate vicinity of an exposed pruning wound. So yeah, being vigilant and pruning in dry conditions is key. Late pruning is a good technique. So there's evidence that shows that these pruning wounds actually heal a lot quicker in warmer conditions. So if you can prune a little bit later, so February, March, again, depending on where you are in the country, that's going to be beneficial. Double pruning is also a technique I don't think is widely used anymore, but with that, you'd make use of your available crew, kind of come through, make kind of a quick pruning cut, maybe a rough and ready one, and then you may get some trunk diseases getting in, and then you'll come back and do a second pruning event to kind of eliminate those a little bit later in the season. The photos on the top, show that these trunk diseases really do infect all parts of the vine, the cord on the trunk, the roots, and the spurs. So if it's heavily infested, you will want to cut out that infected vine. If it's very heavily infected, its longevity is really going to quickly go down. I want to touch upon pruning wound protectants. These are key from the get-go. The idea behind these are effective chemical and biological pruning wound protectants that basically will create a barrier on that pruning wound to stop fungal spores getting in. The key with this is you're going to want to start this as soon as possible in your young vineyards because they can come in very early and they're very slow progression diseases. which kind of has its benefits, but it does mean once you have it, they're just going to slowly get worse throughout the life of the vine, and there's no way to do it, get rid of it unless you can kind of cut it out. Kind of like the spotted lanternfly, identifying the source. So trunk disease is also like a lot of other horticultural plants, nuts, walnuts, almonds are often grown very close to vineyards. So it's kind of adding to that inoculum source. So maybe thinking about different plantings could be key. And then again, kind of the theme, abiotic stress is tied to disease outbreak and severity. So again, general good vineyard management practices all kind of fit together. Okay, so moving on to the last two. So now with mealybugs, so they go through the life cycle, eggs to nymphs and crawlers, and then adults. And they kind of cause damage both directly and indirectly. So firstly, directly, they're depositing honeydew. So they're serving, that substrate we already mentioned about that black sooty mold, which is then going to bring in contamination, infecting bunches. And then very importantly, they do transmit grapevine leaf roll associated viruses. We haven't talked much about viruses today. So I did just want to touch upon this. So this is a big issue in the photo on the right. Was from a Colorado vineyard having quite a extreme outbreak of that virus. And again, so the mealybugs, they can transmit that. So, mitigating them is key. When it comes to mitigating losses, we do have some excellent biological control options. So actually, parasitic wasps, as well as ladybird beetles, are very, very effective. Ladybird beetles actually have earned the name as the mealybug killer. So you can purchase those and release them into your vineyard. You're going to want to make sure, though, that there is actually a fairly good population of mealybugs before you do that to give them a good food source. Otherwise, they're not going to be able to survive too long. Parasitic wasps are great, but they are very susceptible to ants. So you might want to consider, say, planting common vetch between your rows, kind of actually to draw away those ants to let the parasitic wasps do their job. Insecticide treatments as an excellent option. Again, a lot of broad options available there to treat them. And as I kind of mentioned, we really do want to focus on preventing them to your uninfested vineyards and removing the vines of those that are very heavily infected too, especially with the virus. So, with all diseases, monitoring and scouting is key. So you're going to want to peel back your bark and check for those eggs and crawlers in the dormant season. That can often be quite easy to do, and then maybe make applications there. And yeah, lastly, I should kind of say that actually, if possible, having a professional identify your mealybug species, as there are a few key ones, but there can be some important differences when it comes to say applying insecticides based on the species. So if you can get an accurate identification, that may be beneficial as well. All right, so lastly, from my part for today, we're going to talk about powdery mildew. This is obviously a key one for the majority of vineyards, kind of in the United States, particularly here in California. It's always a problem year after year. So powdery mildew, the fungi Uraciphe nectar. So typically this starts, this is definitely an early season issue to try to get on top of as soon as you can. So, first you're going to see kind of yellow chlorotic spots appearing on the top side of the leaves, but then on the underside after that, you're going to start to see the wet mycelium mats spreading. So mycelium will eventually form hyphal strands and it becomes so dense that that's what you can physically see. So on the left is a heavily infected bunch. So that'll be on the bunches on the underside of the leaves. In terms of what generates powdery mildew, it spreads very rapidly between about 70 and 86 Fahrenheit. Fortunately, once it gets a lot more above that, and particularly high UV, it's actually very detrimental to powdery mildew. So that's why it often comes in early in the season and then spreads so quickly. But then maybe not such an issue after that. However, once it's there, it's hard to eradicate. So again, in terms of control, that early season detection is very critical. And then getting in there with that kind of preventative control, as Jake could talk about a little bit as well. So mainly, basing that on the weather. So precipitation is going to, lead to some of those outbreaks. Again, powdery mildew, like all fungal diseases, overwinters a lot on the vines. So getting in there early is going to be key. As we mentioned, for heat, relieving heat damage, shoot thinning, as well as leaf removal. That's going to be key for kind of two reasons. One, allowing you to get a better foliar application in your clusters, as well as actually promoting airflow and reducing that humidity environment in your canopy clusters, which is what mildew thrives on. So we kind of mentioned foliar application. Then lastly, cultivar selection, vinifera versus hybrids. Hybrids do tend to have a bit more of a robust genetic makeup when it comes to dealing with mildew. Perfect. So I think that was that was a lot to cover. Hopefully didn't throw too much there. These are all great topics, and I'd love to spend an hour on each of them. So hopefully there's something in there for everybody. So now I look forward to passing it over to Jake.
Awesome. Thank you so much, Robert. I'm just going to pause here and allow for a little Q&A. I'm not sure, Danny, if this is a question or just a statement. Using high vigor rootstocks may help to grow thicker, stronger canopy. However, grape quality tends to be lesser than less vigorous rootstocks. So just a comment.
It's a good observation, certainly with the comment on new typer as well. So Danny's already similar with the trunk diseases. They can also under extreme conditions cause that trunk to split as well. So
I hand it over to Jake now. Jake, you should have control of the slides.
Okay, good afternoon, guys. I assume everybody can hear me all right.
Yes, we can.
All right. My name is Jake Wiley. I'm the territory sales manager up here in the Chico area, which is pretty close to Sacramento. I cover down into Stockton, Lodi area and up into the Napa Valley, so we get around quite a bit. It's going to let me go. All right, so this is a quick overview of ProFarm's grape portfolio. We've got our bacterial disinfectants, plant health, and insecticides over here. There is one product here not listed. It is Haven, which is our sunscreen. I will briefly talk about it at the end. But everything here, it's noted, is organically labeled and residue exempt with 0-day pre-harvest intervals and 4-hour re-entry intervals. Excuse me. Most of these have minimal PPE. There is a little bit of nuance with your JetAg, Invenerate, and Grandivo, but they're all great options as well. If you've got like a tasting room or something like that where you don't want large Tyvek respirator type suits walking around and making people uncomfortable. So there are good alternatives on that end as well. So let's get started here with JetAg. JetAg is a PAA type product, which essentially is a combination of hydrogen peroxide and acetic acid. When you combine these two products, they form a new product called peroxacetic acid. Now, peroxacetic acid is essentially an oxidizer. It's got 100 times the oxidizing power as commercial grade hydrogen peroxide, and it's more stable. Hydrogen peroxide tends to break down when it's exposed to air. PAA does not do this. It actually stays fine up until it actually dries and is gone off the plant. So this is also why that it is residue exempt, as it's gone once it evaporates off. Now, oxidizing is an interesting word. You might be more familiar with it with regards to rust and metal, but it's essentially a broad term that describes taking an electron from something. So what happens is when this PAA product comes into contact with the cell wall, whether it's bacteria or a disease or anything like that, it steals an electron from it, and essentially it bursts the cell wall, killing the organism. Now, PAA was originally designed as a sanitizer for water treatments, line cleaning, or tank cleaning, such like taking one tractor from a field and taking it to another. You could give it a PAA scrub down, essentially cleaning that tractor. But we've discovered is that this can also be used in a IPM program, essentially as a leaf sterilization or a foliar fungicide in that kind of term. So we'll talk a little bit about what it is. Application rates are going to be a gallon per 100 gallons of water, or 1% volume to volume. Now, it is also a irrigation water treatment, so you can drop that use rate down to 1 ounce per gallon of water, much more cost efficient for a treatment system into your irrigation or drip line. We've already talked about the active ingredients here. You've got your 5%, which is the regular JetAg, and then the 15% is your oxidation or jet oxide. Oxidation of cells, but what happens here is because it's a broad spectrum, we're getting not only fungus, but we're getting bacteria, algae, and diseases that might be on the leaf surface. So what ends up happening is we're scorching the surface of the leaf. It's not penetrating the cells because they have a cell wall that's stronger than a typical bacteria. but you're getting a sanitized leaf surface. Now that we have a leaf surface, let's see what we can do about actually recolonizing that with something better. Stargus is our isolate of Bacillus amylocophaceus, specifically strain 727. There are a lot of other bacilluses on the market. We're going to talk about this one today. They all kind of have different strengths and weaknesses. This is a preventable fungicide, so it needs to be on early. It's labeled for foliar and soil-borne diseases. The label rate is 1 to 4 quarts, but typically we recommend going out at 2 to 4 quart application rate. Typical interval is going to be about 7 to 10 days. Now, everything labeled here on the bottom is actually on the Stargus label, but it definitely shines on your botrytis or gray mold type diseases. We'll talk a little bit about how this actually works. So when we ferment Stargus in the vat, what ends up happening is some peptides are produced in that process, and those are what are actually preventing pathogen infection and or reproduction. Now, Stargus comes with a ISR and SAR symptom to it as well. A lot of other bacilluses on the market, this is actually what they're producing and how they're affecting the plant. Now, it's definitely a symptom of putting out Stargus, so you get that benefit as well. But really where Stargus shines at is it recolonizes that plant surface, and it grows in and around the cells and essentially creates a protective shield or wall that stops anything else from getting in or colonizing on that leaf surface. And let's take a look at that. This is a petri dish that we inoculated. Stargus is in the square up top, and we inoculated gray mold here on the bottom. As you can see, if you look at the square, it has grown out from the original inoculation point. So this is showing how Stargus does in fact colonize that leaf surface and grow out. And then also the gray mold has grown pretty much unchecked all the way up until when it encounters that Stargus. And then it essentially hits a wall. And what it's doing is it's running into those peptides, and it can no longer reproduce and continue to grow. So you can see why this product works very, very well preventatively. If you've got this shield on your leaves, you're never going to get anything that's getting in there and actually colonizing and getting into that leaf surface. But if it's already there, then you're going to have a hard time getting rid of it. And this is where it comes back into, let's take our Jet Ag, which is our leaf sanitizer, and mix it with our Stargus. Now we get the best of both worlds. So what ends up happening here is the Jet Ag cleans off that leaf surface, and the Stargus comes in right behind it and grows pretty much unchecked. No competition or anything across that leaf surface, and creates that wall. Now, the first thing that always comes up here is, well, what about Jet Ag killing the Stargus in the tank? And we've done a lot of studies with multiple hours in the tank with them together. And essentially, we've boiled it down to this. We can guarantee you that if you mix Stargus and Jet Ag and you spray it within 1 hour of that tank mix, you are not going to affect the colonization rate of that Stargus. So we've talked about one option here. We can move into our ISR product, which is Regalia. It is an extract from giant knotweed. Same label rate, 1 to 4 quarts per acre. Typically recommend staying up in that 2 to 4 quarts. You can drop it down to 1 if you're mixing it with like a strobilurin type product. The plant health boost aspect of Regalia can really add to a strobilurin fungicide program. It's going to carry your regular caution style warning on it. It's brain fast in an hour. It does have a spreader sticker in there just to stabilize it in the jug. We recommend throwing a little bit in there with it, but not get overboard with it because there is something in there already. Now, SAR and ISR, it's a term that gets thrown around a lot. Let's talk about what it actually is. So if you take a look at my cell here in the corner, when Regalia hits the leaf surface and enters into the cells, we have this induced systemic resistance or systemic acquired resistance that's created. And what ends up happening is it's kicking the plant's immune system into gear. It creates antimicrobial compounds on the leaf surface that actively repel and kill pathogens that are there. But it's really more of a side product of that. The best part of this is when it goes into the cell walls, it moves a little bit deeper in, it's going to trigger the creation of producing more lignin, which is what gives us stronger cell walls in plants versus humans. So this inhibits pathogen penetration and improves the structural integrity of your cell. And we see this a lot with like our rice and wheat crops that we do this in. The fields that get treated with regalia have a lot less lodging and falling over before harvest at the end of the year. And that's because their stalks are stronger and thicker than the fields without regalia in them. It's much harder to see that in a vine, vineyard, because you've got stronger, taller plants, but it is happening out there. And lastly, as it gets into the cell, it triggers the production of chlorophyll A and B and other phytohormones. Essentially, what you're doing is you're creating about 20-ish to 25% more chlorophyll for a short time in there. It improves photosynthesis, which improves, obviously, energy and its ability to fight off abiotic stresses such as cold and heat. So let's put this all together for us. So we put a little short program together here for you. Regalia is a thick black product that can leave a touch of staining on your leaves. If you actually test that black dot, there's no more regalia in it. It's residue exempt. You can't even test for it. But it can be a little dot there. There are people that use regalia up and through harvest, no problem. But for simplicity's sake, this is how we recommend you do it. Use regalia up front, either in rotation with your sulfur or at reduced rates. A lot of people mix them together at lower rates and get equal, if not better, control that way. And they're also reducing their sulfate load on the front end. It is important to note that sulfur can be a little hot on plants, especially depending on different varieties. So be careful with how much you're mixing and the combination. Up until bloom pre-close, that's when we recommend switching over to Stargist Jet Ag, as it is our really gray mold product. And it doesn't leave any staining or any marking on the plant whatsoever. So it's very safe to use up through until harvest. A little data here for you just to show we're not talking about nothing. If you take a look on the left, we have our axis here with the percentage of control. And then on the bottom is our date. So this is with powdery mildew done at UC Davis right here in California. If you know, some of these years jump around a little bit on the bottom. It's not because we're cherry picking the data. This is an ongoing study that UC Davis does every year. And essentially what it boils down to is some years they include this in the study and some years they don't, such as 2018 here on the bottom. It's been mentioned twice, and that's just because they used this twice that year. So we have a history of continual control of powdery mildew in the grapes in the California area here. Before we jump into the insecticide portion of this, are there any quick questions on our biofungicides?
There was a question about if the products were labeled in greenhouse, and Taylor answered that.
Perfect.
They are.
We're a little short on time. I'll let Taylor continue to chip away in the comments, and I'll continue.
There's one quick question. Does it enter leaf surface through diffusion or active transport?
If it's regalia, it's going to be active transport. It gets brought into the cell.
Okay, let's keep going, and then we can answer it at the end too. Okay.
All right, Venerate is our flagship insecticide. It is Burkholderia rhinogenesis, which was actually discovered in a Buddhist temple in Japan, weirdly. It is the same 1 to 4 quart label rate. 2 to 4 quarts for alone use. And then we've seen we're starting to do a lot more studies with Venerate at a 1 quart rate in your conventional chemistries, specifically with miticides that are a little bit more tired. Adding just a quart of Venerate to it can really spruce up the efficacy of those products. Typical caution and application is foyer. We do have a version of Venerate that's labeled for chemigation. It's under Magistine. So Venerate's great on aphids, mites, thrips, all of your mealybugs for the most part, whiteflies, leafhoppers, and it really, really shines on lepidopterans. So let's talk about a little bit how this Venerate works. It is locally systemic like regalia, meaning it gets into the top couple layers of cells. Death typically occurs within 4 to 7 days after feeding. Chewing insects, also along with piercing, sucking, end up ingesting Venerate. It's a gut buster type product. It's not necessarily BT, but it acts similar to it. It is important to note, though, that takes a little bit for it to die. So you should go out and scout these fields and make sure that the insects are walking around sick or falling off. It's not going to happen immediately, like you might see with some other products. So Venerate is a broad spectrum pest killer. It happens on lepidopterans and mites, aphids as well. But you might be thinking, well, what about our beneficial insects? I'll show you a study later, but we've seen a lot of insects that continue to survive and thrive in areas where Venerate has been applied. Now, we do have another insecticide. It is Grandivo. It is a chromobacterium. This is a dry formulation. 2 to 3 pounds is the label rate. Same application, soil and foliary. It's got a caution word. It's got a very similar label to Venerate. Really, the major difference in this product is it's got a active pest repellency factor to it. So if you go out, especially with your mobile insects like thrips and whiteflies, a field treated with Grandivo is much less likely for insects to actually come into, as they don't like being around on it. It is important to note that for coverage, though, because if you've got suckers or something like that on the bottom of your tree, if Grandivo gets sprayed on the top, your mobile insects are going to jump down and stay on those suckers and then come back after it degrades. So proper coverage or sucker control is pretty important for Grandivo. Now, also a question that comes up is Venerate or Grandivo, which one should I use? And a lot of times it just depends on which one you've used and are more comfortable with. So my typical recommendation is going to be rotate them every 7 to 10 days. And this has a lot to do with the way they act. They have a similar mode of action, but it's not necessarily the same at a molecular level. So what we've seen is if you mix Venerate and Grandivo together, you don't get a boost of efficacy, so to say. It's relatively the same. However, if you rotate them between different sprays, you also don't have this resistance issue, or they don't get tired, because essentially they are doing different things on the insect. The rotation is good enough to keep both the chemistries alive. So depending on what you're going after, a rotation is the best way to use these products. And lastly, to note on these two products is their biggest downfall is they are not ovicidal, meaning they're not going to be killing the eggs. So anytime you can get an oil in with either of these products or mix it with something that's going to kill the eggs, you're going to get a huge boost in efficacy as well. Here is the study I was talking about. This was done with both Grandivo and Venerate with the Vineland Research Institute. Is our first study here. It was done at 96 hours of exposure at 3 times the maximum label rate, and there was no statistically significant mortality with any of the labeled predatory mites here. Secondary study we did at 24 hours, 3 times the maximum label rate, no significant mortality between any of the beneficial insects listed here. Now, lastly, a little bit of data to back this up. Vine mealybug is the major pest that we're going after. You probably recognize this AI that's in this chart. We've got cluster damage on the left and our active ingredients on the bottom. As you can see, A through D is our applications. So it means that there was 4 applications done here. 1 quart of Venerate was not quite as good, but stood up to it, and 2 quarts was just as good as our grower standard here. I would recommend staying away from the 1 quart. It did well here, but typically we see better, more consistent results with 2 quarts all the way up into 4 quarts. Similar with Grandivo, a similar study on mealybug. We've got number of mealybug nymphs per 5 shoots on the axis over here. And these were evaluated at 4 different times. And these were sprayed also 3 times. As you can see, Grandivo actually outperformed the grower standard here as well. Now, lastly, I'll wrap up to get you guys out of here a little bit over time. Haven is our heat stress manager. It is the only product that we have that is not organically labeled, but it is also residue exempt. It's 4-hour reentry and 0-day pre-harvest, much like our other products. Now, the active ingredient at Haven is, if you look in Agrian, it just says alcohol on it. But what it really is it's a derivative of a sun or a coconut oil type product. It's got a UV reflector in it, and that's actually a side product of the active ingredient. It's something we use to stabilize it when it's being made that actually has UV reflection in it. So you're definitely going to get a little bit of boost or life out of your typical insecticides or fungicides that might be up there. But what the active ingredient Haven is doing is it's affecting the stomata regulation of the plant. Now, Rob mentioned that when things get really hot out there, the stomatas close, stops transpiration, and the plant kind of goes into an almost pseudo hibernation because it's so hot. Now, what Haven does is it opens up those stomata, increases transpiration, and regulates the water use of the plant. So not only are you getting continual water up through the plant, you get better use efficiency out of it. Now, what comes along with better water use efficiency and more liquid going through the plants? Typically, we're seeing more nutrient uptake because of this. And in turn, we're seeing better yields with crops that have Haven on it, just essentially because the plants are working better for longer. Now, this product is actually clear. It's not a kaolin clay type product, so it's not going to make a mess out of everything. You don't have to wash any fruit when you're done with it, and it's not going to clog your nozzles. This is our typical art of use for it. Haven is a product that needs to be put on before a heat event. It shouldn't be done in response to it. Typical application is going to be at least 3 times a year, and then continuing after that as needed. As you can see, grapes are the top chart here. I have melons and tomatoes on here as well, just for reference. So in summary, Regalia is used best early season, 2 to 4 quarts per powdery mildew. Jet Ag at 1% volume to volume for curative. It can be used year-round. It improves the colonization of Stargus. It is also take mix compatible with Regalia. I forgot to mention that. It can be a little finicky with heavy metals such as copper, zinc, and magnesium, but it's not really like a phyto kill the plant type response in that note. Stargus is best used late season. It's great on botrytis. We recommend the 2 to 4 quart use rate here. And then lastly, for Venerate and Grandivo, they're best used in rotation, and Venerate can be used at a 1 quart late to improve your conventional program. All right, our gift giveaway here. Anybody answers quickly, where do we recommend switching from Regalia to Stargus Jet Ag combination? Like a pre-close right here. The first comment got it. So we do recommend switching at pre-close. We'll have someone respond in the comments and get some stuff to you. All right, Angela, that's all I got.
Great job, Jake. Thank you. So Jake just switched to this slide here. So based on where you're located, this is another great slide just to screenshot or take a photo of. These are your technical contacts for ProFarm Group throughout the United States. This is also available on our website as well. So you can always go to profarmgroup.com and then just click on Talk to an Expert, and you'll find these contacts as well. Can Jet Ag be mixed with sulfur application?
Sulfur is a no-no with Jet Ag, I believe.
Right. Are there any final questions today? Robert, Jake, I really appreciate all the time and expertise you've shared today. Thank you so much.
Thank you. And thank you to the excellent attendees. Great questions today. And thank you for staying engaged.
Thank you again for joining us on our webinar, and please stay tuned. We have our YouTube channel. You can just search profarmgroup.com or go to our URL, our website, where you can find many more webinars. So if you work with a variety of crops, we try to do these educational webinars on a number of crops. And of course, if you have any feedback or suggestions for future webinars, I always love to hear. We're always looking to be better and provide more helpful resources for the industry. So thanks again for attending, and thank you, Robert and Jake, for putting on a great webinar. Goodbye, everyone.